Hello everyone, welcome to Kaur's classes. I'm Dr. Mandeep Kaur and today we are going to discuss TCA cycle. So Krebs cycle, also known as TCA cycle, tricarboxylic acid cycle. It is used to oxidize the pyruvate formed during the glycolytic breakdown of glucose into carbon dioxide and water with the release of energy. So it also oxidizes acetyl coenzyme A, which arise from carbohydrates, fats, and proteins. So, in addition, this cycle provides precursors for biosynthetic pathways. So, this cycle operates in mitochondria of eukaryotes and in the cytosol of prokaryotes. It is an amphibolic cycle because it is both catabolic and anabolic. It breaks down certain molecules and it builds up certain molecules. So, it is an amphibolic cycle. Now, coming to the cycle, it forms the central path of three-stage processes which oxidizes organic fuel into carbon dioxide with the release of energy. The first stage is oxidation of fuel molecule to acetyl CoA. So, in the first step, what happens? The major source of energy is glucose. So, this glucose is converted into pyruvate with the help of glycolysis, and pyruvate it get converted into acetyl CoA with the help of an enzyme known as pyruvate dehydrogenase. So in this step this whole reaction is a linking reaction of glycolysis to Krebs cycle. So in this particular reaction, what happens? There occurs release of carbon dioxide. The three carbon pyruvate it get converted into 2 carbon acetyl molecule and with the release of 1 carbon dioxide. And the coenzyme A it get attached to acetyl molecule and it forms acetyl CoA with the help of enzyme pyruvate dehydrogenase. So in this step there occur a release of carbon dioxide and reduction of NAD positive to NADH. So this reaction is oxidative decarboxylation. So this pyruvate dehydrogenase enzyme, it's a complex which is made up of 3 enzymes and 5 coenzymes. The first enzyme is pyruvate dehydrogenase, also known as enzyme 1. Second enzyme is dihydrolipoyl transacetylase, abbreviated as enzyme 2, and dihydrolipoyl dehydrogenase, abbreviated as enzyme 3. So these three enzymes, they have different cofactors associated with them. They have different subunits in prokaryotes and eukaryotes and different functions. So first enzyme, pyruvate dehydrogenase enzyme, it has thymine pyrophosphate as cofactor, which is also known as vitamin B1. So this pyruvate dehydrogenase enzyme 1, it has 24 subunits in prokaryotes whereas 30 subunits in eukaryotes. Its function is oxidative decarboxylation of pyruvate. Second enzyme, dihydrolipoyl transacetylase, it has lipoid and acetyl coenzyme A as cofactors, and there are 24 subunits in prokaryotes and 60 subunits in eukaryotes. Its function is to transfer acetyl group to coenzyme A. And third enzyme, dihydrolipoyl dehydrogenase, also abbreviated as enzyme 3, it has FAD and NAD positive as cofactors. And in both prokaryotes and eukaryotes, there are 12 subunits of this enzyme. And the function of this enzyme is to regenerate oxidized form of lipoid. Now coming to stage 2, the citric acid cycle. So this cycle carries out the oxidation of acetyl group from acetyl coenzyme A to carbon dioxide with the production of 4 pairs of electrons stored in reduced electron carriers NADH and FADH2. So basically there are 8 steps in this cycle. The first step is condensation, in which there occur condensation of oxaloacetate and acetyl coenzyme A, which combine to form citrate. So oxaloacetate is a 4-carbon compound, whereas acetyl-CoA is 2-carbon compound. So in the first step, these two molecules, they combine to form a 6-carbon compound known as citrate with the help of an enzyme known as citrate synthetase. In the second step, there occur isomerization of citrate to isocitrate with the help of enzyme aconitase. In the third step, there occur oxidation of isocitrate to alpha ketoglutarate with the help of an enzyme isocitrate dehydrogenase. You always have to remember that whenever there is an oxidation reaction, the enzymes which are into action are dehydrogenases. In the next step also there occur oxidation of alpha ketoglutarate to succinyl coenzyme A. Also there is release of carbon dioxide, so this is oxidative decarboxylation. So step 3 and step 4 again are oxidative decarboxylation. So once succinyl coenzyme A is formed, there occur cleavage of the acetyl CoA group and it leads to the formation of succinate, and the enzyme into action is succinyl coenzyme A synthetase, and there is release of GTP. The GTP combines with the inorganic phosphate and forms this GTP. In the sixth step, again there is oxidation of succinate to fumarate with the help of enzyme succinate dehydrogenase, and there is formation of FADH2. In step 7th, there is addition of a water molecule. So the step is hydration, and there is formation of malate from fumarate with the help of an enzyme fumarase. So the malate so formed, it oxidizes into oxaloacetate with malate dehydrogenase and formation of NADH. So again, basically there are 3 steps in which there is production of NADH. Step 3, where isocitrate get converted into alpha ketoglutarate, one NADH is formed. Step 4, when alpha ketoglutarate get converted into succinyl coenzyme A, again NADH is formed. And step 7, when malate is converted into oxaloacetate, it forms one NADH. So beside these 3 NADH, there is formation of FADH2 and GTP. Now coming to bioenergetics of TCA cycle, as I have just discussed, that there is production of 3 NADH at step 3, 4 and 8, whereas there is production of 1 FADH2 molecule and production of 1 GTP. So overall 3 NADH2, which leads to the formation of 1 NADH is equalizes 3 ATP. So this forms 9 ATP. 1 FADH molecule, it leads to the production of 2 ATPs. So 1 FADH2 produces 2 ATPs. So 9 ATPs plus 2 ATPs and plus 1 leads to the formation of 12 ATPs overall. So thanks for watching.